Let us see Nutrition and Dietetics Part 2. See, biotechnology. Biotechnology is the science of applied biological process. In other words, it is science of development and utilization of biological processes, forms and systems for the benefit of mankind and other life forms. Okay. The term biotechnology was coined by Karl Erke, a Hungarian engineer, in 1919. Okay, this point you have to keep in mind. Okay, the term biotechnology was coined by Karl Erke, a Hungarian engineer, in 1990. Okay, biotechnology has been extended to include any process in which organisms, tissues, shells, organelles, or isolated molecules such as enzymes are used to convert biological or other raw material to product of greater value. Green manure. This topic is green manure. Green manure is obtained by the collection and decomposition of green leaves, twigs of trees, shrubs and herbs growing in wastelands, field burns, etc. Okay, green manure improves the soil structure, increases water holding capacity and decreases soil loss by erosion. It also helps in reclamation of alkaline soils and reduces weed proliferation. It's a manure obtained from uncomposed and decomposed green material derived from leguminous plants. For example, sun hemp, okay, daincha, sesbania. So all this. So what is green manure in the sense? Green manure which is obtained by the collection and decomposition of green leaves, twigs or trees, shrubs and herbs growing in wasteland or in field barns. Okay, those leaves are collected and they put in a pit. Okay, put in a pit and it is covered with the sand or soil. Okay, after few days what happens now? The bacteria acted upon this bacteria as well as the fungi, okay, the decomposes acted on this leaves and convert these leaves into a green manure, okay, organic manure. So these manures will be then dug it and it will be supplied to the or put into the paddy field. Okay, this is called as green manure. So how the green manure prepared in the sense? Green manure are nothing but the leaves as well as the twigs which is cut out from the trees or the shrubs or herbs. Okay, all those things are collected in a pit and just it is covered with the soil. Then after 1 or 2 months later, all the decomposes which present in the soil decompose all the greens, okay, all the waste of that is the plant waste, okay, and it convert into an organic manure. That manure is called as green manure. Okay, this manure is called as green manure. Then it will be dig and just supply to the paddy fields. Okay, and this is very good for the plants and it produces a very good harvest we get. And frankly to say, it improves the soil texture. Okay, the green manure improve the soil texture as well as with the retention of water will be more in the green manure. If you put the green manure to the paddy field, the soil texture and the water retention capacity will be more. Okay, next one, nanotechnology. What is nanotechnology? Nanotechnology in agriculture. Currently, nanotechnology provides different nano devices and nanomaterial that have a unique role in agriculture. For example, nano biosensor is used to detect moisture content and nutrient status in the soil. This point you have to keep in mind. Nano biosensors. Okay, nano biosensors. So what are nano biosensors in the sense? It is used to detect the moist content as well as the nutrient status in the soil. For example, see, with this sensor we can find out the status of the soil, and we supply which nutrient is necessary for that particular plant. So this will be very useful. Nano biosensors. Okay. Next one, nanotechnology can offer nano fertilizer for efficient nutrient management, nano herbicides for selective weed control in crop field, nano nutrient particles to increase the seed vigor, nano pesticides for efficient pest management. Okay, so the nanotechnology is giving how many different things in the sense: nano fertilizers, nano herbicides, okay, then nano nutrient particles and nano pesticides. Let us take the first one. Nano fertilizers are very useful because it is the efficient nutrient which supplies to the plants, okay, or the crops. Then next one, take the nano herbicides. These nano herbicides are the selective weed control, okay, selective weed control, nano herbicides. And the nano nutrient particle which trigger or which increase the seed vigor, okay, nano nutrient particles. And nano pesticides for efficient pest management, okay, the pest will be managed very efficiently by the nano pesticides, okay. Then hence the nanotechnology have greater role in crop production with the environmental safety, ecological sustainability and economic stability. See, by studying the objective question answers without knowing the subject, you won't retain in your mind for a long. Okay, so whatever you're studying or learning, just go through the subject properly, then it will be in your mind for a long term. Okay, so it will be stored in your memory in a long term memory. Okay, instead of studying the options, you can study the subject in a proper way. Now next one is bioremediation. So what is bioremediation? It is defined as the use of the microorganisms or plants to clean up the environmental pollution. Okay, so environmental pollution should be cleaned up then and there, okay, by the microorganisms. So it's an approach used to treat waste, including the wastewater, industrial waste and solid waste, okay. And bioremediation process is applied to the removal of oil, petrochemical residues, pesticides or heavy metals from the soil or groundwater, okay. So this is called as bioremediation. So some remedy we have to take to remove the pollution from the land, water, and what are the pollutants in the sense the petrochemical residues, pesticides and heavy metals from the soil or the ground or on the in the ocean, okay. So we have to remove, that is called as bioremediation. Biofertilizers. So what are biofertilizers? Biofertilizers are the substances that containing living microorganisms when applied to seeds, plant surfaces or soil colonize the rhizosphere or the interior of the tent plants and promote growth by increasing the supply or availability of primary nutrient to the host plant, okay. So biofertilizer means what? The substances is containing living microorganism. This is the point, okay, which contain the living microorganism, that is called as biofertilizer. When it is applied to the seeds, plant surface or soil, colonize the rhizosphere or the interior of the tent plant and promote the growth of the increasing the supply of availability of primary nutrient to the host plant, okay. Bioframing, also known as molecular forming, okay, is the production and use of transgenic plants genetically engineered to produce pharmaceutical substances for use of human beings. This is also called molecular forming or forming. These plants are different from medicinal plants which are naturally available. See here, transgenic plants, right? So what is this transgenic in the sense? It is by using the biotechnology, they They remove certain undesired gene. Okay, see, for example, this is the plant. It's a dwarf plant and it is a very strong variety. Okay, very strong variety, but it is very dwarf. Okay, and less yield. This is less yield. Just imagine, this is less yield. Okay, and whereas another variety which is a long one, more yield, but it is susceptible, that is, which dries up very quickly. Okay, very quickly in drought condition. So what the scientists do in the sense, they take a necessary gene from here and they transplant the gene here. Okay, by the method called genetic engineering, they remove the gene and they incorporate another gene into that. So here what happened, what is the result of this genetic engineering, in the sense they can withstand in the drought condition and they yield more. Okay, they yield more. So this is called as transgenic plants. Okay, transgenic plants. So in such cases we can get a very good crop with a good yield. Okay, so bioprospecting. What is bioprospecting? Bioprospecting is the process of discovery and commercialization of new products obtained from biological resources. So bioprospecting may involve biopiracy, in which indigenous knowledge of nature originating with indigenous people is used by others for profit without authorization or compensation to the indigenous people themselves. Biopiracy. What is biopiracy? It can be defined as a manipulation of intellectual property rights law by corporation to gain exclusive control over national genetic resources without giving adequate recognition or remuneration to the original processors of those resources. Examples of biopiracy include recent patents granted by the US Patent and Trademarks Office to American companies on turmeric, neem, and most notably the basmati rice. Okay, all three products are indigenous to the Indo-Pak subcontinent. Green fluorescent protein, GFP, green fluorescent protein. The green fluorescent protein is a protein containing 238 amino acid residues of 26.9 kDa that exhibits bright green fluorescence when exposed to blue to ultraviolet range. Okay, the range is 395 nm. GFP refers to the protein first isolated from the jellyfish Aquaria victoria. Okay, GFP is an excellent tool in biology due to its ability to form internal chromophore without requiring any accessory cofactors, gene products, enzymes, or substrates other than molecular oxygen. Okay, so what is this? May be a question. So green fluorescent protein, green fluorescent protein. Okay, so this protein contains 238 amino acid residues. Okay, residues of 26.9 kDa. Okay, kDa into that exhibits bright green fluorescence. Okay, when exposed to the ultraviolet range. Where they found, in the sense they found in jellyfish Aquaria victoria. Okay, and it is an excellent tool in biology due to its ability to form internal chromophore without requiring any accessory cofactor, gene product, enzymes, or substrate other than molecular oxygen. Let us see cryopreservation. Cryopreservation is also known as cryoconservation. Is a process by which protoplast cells, tissues, organelles, organs, extracellular matrix, enzymes, or any other biological materials are subjected to preservation by cooling to very low temperature of minus 196 degree centigrade using liquid nitrogen. Okay, so cryopreservation. What is cryopreservation? It is a process by which protoplast. Okay, we all know what is protoplast. Cells, tissues, organelles, organs, extracellular matrix, enzymes or any other biological materials are subjected to preserve by cooling at temperature. What is the temperature? Minus 196 degrees centigrade. Okay, by using liquid nitrogen. Okay, so cryopreservation, keep in mind. This is very, very important. Cryopreservation. Okay. Now, at this extreme low temperature, any enzymatic or chemical activity of the biological material will be totally stopped. Okay, why we are preserving in minus 196 in the sense the total activity of the cells will be stopped. Okay, will be stopped, and this leads to preserving the material in dormant status. Okay, they go in inactive state. Dormant state in the sense inactive. The cell goes in inactive, but the cell is alive. Cell is alive, but it is in dormant status. Okay, later whenever it is necessary, that will be taken out and will be utilized. When we bring this into the room temperature, again all those will be in active status. Okay. Next one, application of biotechnology. So wherever this biotechnology is utilized, that we are going to see one by one. This science has an invaluable outcome like transgenic varieties of plants, transgenic cotton, rice, tomato, tobacco, cauliflower, potato and banana. Okay, so all these the genes are changed and brought out a very good crops. Okay, that is what we call hybrid varieties. Okay, now the development of transgenics as pesticide resistance, stress resistance and disease resistant varieties of agricultural crops is the immense outcome of the biotechnology. Otherwise the pests will come and completely destroy the crops, and so much loss will be faced by the farmers. That will be stopped by this method. The synthesis of human insulin and the blood protein in E. coli and utilized for insulin deficiency disorder in human is a breakthrough in biotech industries and medicine. This point is also very, very important for the exam. Note it. The synthesis of human insulin, blood protein in E. coli. Okay. These are utilized for the insulin deficiency disorder in human. It's a breakthrough in biotech industries and medicine. So those who the kidney failure people, they are utilizing the insulin by this method. Okay, by biotechnology, we are synthesizing the human insulin. Now, the synthesis of vaccines, enzymes, antibiotics, dietary products and beverages are the products of biotech industries. These are all this synthesis of vaccine, enzymes, antibiotics, dietary products and beverages. All are biotech industries, the products of biotech industries. Biochip based biological computer is one of the success of biotechnology. Genetic engineering involves genetic manipulation. Tissue culture involves aseptic cultivation of totipotent plant cell into plant clones under controlled atmospheric condition. Okay, that is a single cell is able to produce a complete plant or the whole plant. That is what called as that cell is totipotent. Okay. So that also brought by genetic engineering. Then single cell protein from spirulina is utilized in food industries. Single cell protein from spirulina is utilized in food industries. Production of secondary metabolites, biofertilizers, biopesticides and enzymes, all these are All these are by the biotechnologies. Okay, the production of metabolites, biofertilizers, biopesticides, enzymes, human insulin, all are the products of the biotechnology. Now let us see the transgenic refer to an organism. Transgenic means it refers to the organism or cell whose genome has been altered by the introduction of one or more foreign DNA sequences from another species by artificial means. Then it is called as transgenic organism. That organism is completely transformed into another what is that, genetic condition, okay, or generated in the laboratory for research purposes. What is transgene? Transgene is a gene that has been transferred naturally or by any of a number of genetic engineering technique from one organism to another. The introduction of a transgene is a process known as transgenesis, okay, has the potential to the change of phenotype of an organism. So that particular gene may change the phenotype of an organism. So father of Indian medicines. Who are the father of Indian medicines? Let us see. Ayurveda, the father of medicine. Father is Charaka Samhita. Yoga, it is Patanjali. Unani, it is Hippocrates. Siddha, Agastya. Homeopathy, it is Samuel Hahnemann. So thanks for watching this class and share this video to whom preparing for competitive exams and subscribe this channel. Thank you. Thank you so much.